Good day. Today our lecture about microscopic fungi, causative agent of opportunistic mycosis. There are conditionally pathogenic fungi which cause disease in the human body with decreased ability for protection. Patients with compromised host defenses are susceptible to fungi, are referred to as opportunistic fungi. Healthy people, if exposed to fungi, are usually resistant. Causative fungal agents. First, yeast-like fungi, Candida subspecies, Ptolopsis, Cryptococcus, and another species of yeast-like fungi. Filamentous B groups, filamentous fungi which cause infectional disease, Aspergillus, Mucor, Absidia, Rhizopus, and another species of filamentous fungi, and other fungi, pneumocystis subspecies. I will explain about pneumocystis carinii and pneumocystis eurovinii. Pneumocysts and pneumocytosis. Pneumocysts belong to blastomycetes class. Yeast fungi which multiply by budding. Causative agent pneumocystis carinii and pneumocystis eurovinii. These are conditionally pathogenic microorganism and extracellular parasite characterized by strict tropism to lung tissue. Forms of pneumocyst development. First forms is trophozoites. Are involved 1 to 5 micrometers and is irregularly shaped. It is thought to reproduced by binary fission. Second forms of pneumocysts is precysts. The precyst is recognized as an intermediate stage of the sexual phase of reproduction, leading to cyst development 5 to 8 micrometers. Third stage is cyst stage. Can be stained with methenamine silver GMS stain, toluidine blue gimza, and calcofluor white. And the last stage and last forms of pneumocyst is intracystal bodies, or we know like sporozoids. On this slide you can see different stage of formation pneumocysts, different forms of pneumocysts. First and second, it's trophozoids forms. Third, 4 and 5 stage, it's sexual stage or sexual phase reproduction of pneumocysts, and 6 it's cyst contain 8 intracystal bodies. On this slide you can see life cycle of a lot of pneumocytes. Example, pneumocyst eurovinii or Girovici. The parasite enters the lungs in respiratory droplets and gets attached to alveolar epithelium. It divides by binary fission. Some forms a thick wall cyst within which sporozoids develop. When mature cyst ruptures, sporozoids are released, which initiate fresh cycle of infection. You see on this slide stage of our life cycle of pneumocystis eurovinii. First parasite attach respiratory tract, attach alveolarum epithelium, and after binary fission, trophozoid infection stage. After binary fission, forming precyst, developing cyst, forming sporozoid, and sporozoid are released and infected another alveolar cells. Pneumocystis urovini, you see different methods staining of pneumocyst cause pneumocystosis pneumonia, a diffuse intestinal pneumonia. You can see X-rays pneumonia. Yeast-like fungi originally classified as protozoan. Most infections are asymptomatic. Immunosuppressions, for example AIDS, acute immunodeficiency syndrome, predisposes to disease. Diagnosis by bronchoalveolar lavage or lung biopsy. Disc shape is seen on methylene silver stain of lung tissue. You see methylene stain of lung tissue with or with fluorescence antibody. You see, for example, trophozoite forms in lung cells, and quistes. It's another stain method study of microorganism reaction of immunofluorescence, for example, and you see green color of pneumocystis. Pathogenesis. Pneumocystis carinii, pneumocystis jirovii is normally a commensal in the lung, spread by respiratory droplets. 10% of healthy persons have been reported to carry the organism in the lungs. I'll talk about it. Pneumocystis infection is acquired early in life. In immunocompetent individuals, infection is asymptomatic. However, in immunocompromised patients, AIDS, serious life-threatening pneumonia can develop. It's same with this characteristic, same with pneumocystis urovii and pneumocystis carinii. Risk groups of people subjected to pneumocystis infection. First groups, AIDS patients, preterm and weakened neonates, children and adults with immunodeficiency disease, for example, gamma-globulinemia, decreased T cells immunity. Next groups with high risk, patient receiving cytostatic and immunodepressants for a long time, patient with oncological disease, a patient who underwent organs transplantation. For laboratory diagnosis, pneumonia causative agent different strains of pneumocystis, we use specimen bronchoalveolar lavage, lung biopsy or induced sputum are stained, and examined for the presence of cyst or trophozoite inside of alveolar cells and lung cells. For this, we used microscopic technique and use Gimza technique, Gamoris technique, chalcofluoride technique, and immunofluorescence technique for study microorganism, for study trophozoite or study cyst inside of cells. On this slide, you can see, for example, figure A. This Gamori methylamine staining technique. This, for example, reaction of immunofluorescence staining. You see green color of pneumocyst inside of cells. And B, it's Gimza staining for use for rapid identification or tropism forms of the organism. And chalcofluoride fungal cyst wall strains by used rapid confirmation of the presence of the forms pneumocystis calciform white. You see pictures which you see under microscope after chalcofluoride staining technique. For make clinical diagnosis, you can use serological technique for detection of antigens or pneumocystis in the test material, complement fixation reaction and latex agglutination test is also used. And the serological technique you can use for detection of antibodies in the blood serum of patient during disease dynamic immune enzymes analysis. We can use molecular genetic method. We can use PCR for detection genetic material of pneumocystis. And don't forget, pneumocystis carinii cannot be cultured. We can't use nutrient media for cultivation of pneumocystis. For treatment your patient, you will use acute cases of pneumocystis pneumonia. You will treat your patient with 3-metoprim sulfomethoxazole or pentamidine isethionate. It's antibacterial drugs which you use for antibiotics which you use for treat your patients. And next groups of microorganisms which cause opportunistic disease, it's candida and causative agent of candidosis. Classification or taxonomy: family Cryptococcaceae, genus Candida, and a lot of species Candida can cause opportunistic disease, but very often Candida albicans and Candida tropicalis cause disease up 18 to 19%. Candida genus have different shape. You see this shape of Candida tropicalis, Parapsilosis, Cruzea, and Candida glabrata, different shape of Candida you can see on this slide. And about morphological features, first, monocellular formation of an oval or circular extended form. Next, Candida forming pseudogypha, forming blastospores, for example, and forming chlamydospores. You see yeast form of fungi, yeast, budding yeast, multiply like binary fission, and forming pseudogypha, forming chlamydospores, and forming blastospore for multiplication, for reproduction. On this slide you see how you can see Candida under microscope gyphal growth, this invasive stage of Candida albicans. On this slide you can see structure of Candida albicans. I will not explain it because you had time for it. You will have time for it, and I will ask you in our lesson. I told that Candida form two forms, yeast forms and micellar or pseudogyphal forms, micellar forms of Candida. Opportunistic fungal infection. I told a lot of microorganisms, Candida, fungi can cause infectional disease, and on this slide you can see different causative agent and different characteristic of disease which cause this microorganism, fungi. On this slide you can see different form of fungi, yeast fungi, micellar form of fungi. You can see different clinical symptoms, candidosis for example. And tinctorial features. Staining technique for study fungi under microscope. We use simple technique, methylene dark blue, Grams technique, dark blue, violet color of fungi, and Gimza technique, bright red color of nucleus, blue color of cytoplasm host cell. Pathogenesis: superficial, cutaneous or mucosal candidosis, mucocutaneous lesion, oral thrush vulvovaginitis, balanitis, conjunctivitis, keratitis, cause systemic candidosis, intestinal candidosis, bronchopulmonary candidosis, septicemia, endocarditis, and another infection called systemic candidosis, chronic mucocutaneous candidosis. On this slide you can see like commensal state, yeast state, attached normal epithelial cell, and forming pathogenic state, Giffal stage, or with pathogenic properties which increase immune response, active macrophage, active phagocytosis, active neutrophils, and increase different type of adaptive immune cells and increase different cytokines during immune response. Cultural features. For cultivation of Candida, we can use nutrient media. For example, we use simple nutrient agar, meat peptone agar with glucose, blood agar, and the main agar which we use for cultivation of Candida is Saburomedium. On surface Saburomedium, Candida form R and the S form colonies are creamy white with yeast odor. Species of Candida can be identified depending on fermentation and assimilation of various carbohydrates. On this slide you can see nutrient media chromogenic agar for cultivation of Candida, chromogenic Candida agar, and Saburo agar, and you will see different type of colony, S form colony, R form colony on surface agar. Chromogenic agar give you possible see different color of microorganism because different microorganisms have different fermentative activity, enzyme activity. For example, on this slide, S form colony forming Candida. This microscopic examination of Candida albicans. You can see Candida albicans on surface Saburo agar. On this slide you can see how grows Candida albica on surface Saburo agar with dextrose media, and blood agar you can see white colony and yeast form of fungi under the microscope. Identification. The following tests are done to differentiate Candida albicans from other species. Candida albicans is identified by the production of germ tubes or chlamydospores. A. Germ tube test. Candida albicans has the ability to form germ tube within 2 hours when incubated in human serum at 37 degrees. Chlamydospores develop in a nutritionally defensive medium such as corneal agar at 20 degrees. They can be seen at the end of the pseudogypha. And the C type of identification is carbohydrate fermentation and carbohydrate assimilation test. A test and B test I show, for example, forming yeast-like forms and filamentous forms. On this slide you can see yeast cell with germ tube, and after cultivation in 20 degrees temperature you can see chlamydospores. Antigenic features of Candida is glycoproteinic antigen, serotype A, height level of Manan, height virulence, and by glycoproteinic antigen classified by serotype B, decreased level of mana, low virulence, and contain surface proteins antigens. Pathogenicity factors, adhesion factors, surface proteins. Next, integrinoform structure exhibit tropism to the cells of mucous membrane covered with multi-layer flat epithelium. Molecular involved in latinoform contact, superficial glycoproteins, polysaccharides, and monoproteins. Second, factors of invasiveness: phospholipase, proteinase, collagenase, hemolytic enzymes, and morphological transformation from yeast form into a Giffel form help of Candida invasive entry inside of our cells. Next groups pathogen factors of pathogenicity or virulence factors is factors of aggression. It's receptors binding the product of complement degradation and as a consequence blocking attachment of neutrophils. And third group's toxin, candidotoxin, toxin glycoprotein, low molecular toxin, and lipids, this toxic structure of candida. 5 levels of interaction between candida and target cell we know. First, carrier state. Second, superficial candidosis. Third, ulcerative lesion of the mucosa and the skin. Fourth stage, granulomatous visceral candidosis. And the last stage interaction, candidosal sepsis. Candida carrier state can be transitory, short-term, long-term, and chronic. This depends the time of carrier state. Penetration of candida triggers protective reaction. In lesion of surface, the fungi impair the integrity of barriers. Macrophages catch them but are enabled to destroy candida. Cut down proliferation on mucous membranes, stimulate production of secretory immunoglobulin A. Neutrophils can phagocytose and destroy blastospores. They damage pseudomycelium. Nevertheless, parts of microorganism remain viable due to an incomplete phagocytosis. Candida interact with T helpers. Cellular immunity is formed and local granuloma. I show pictures how pathogenic stage Giffel forms of microorganism interact with macrophage, active phagocytosis, interact with neutrophil and then another cells of adaptive immune systems. And technique of laboratory diagnosis. Specimen is swab, scraping from superficial lesion, blood, spinal fluid, tissue biopsies, urine, exudate, and the material from removed intravenous catheters. For identification and for study, for make clinical diagnosis, we use microscopic technique. We used crush drop specimens in Lugol solution, and we used staining technique, simple staining technique, methylene dark blue, Gensen violet, and complex staining technique, Gram technique. Gram staining for pseudo-Giffel and budding cells, it's gram positive microorganism. You see yeast form and pseudo-Giffel cells. As candida can be seen on normal skin or mucosa as well. Only it's a wouldn't present is of significance. Demonstration of miscellaneous forms indicated colonization and tissue invasion, and is their form of greater significance. Skin or nail scrapings are first place. Second groups of my laboratory diagnosis is mycological technique. We used nutrient media, sub-agar for cultivation of Candida albicans, and we will cultivate microorganisms of a sub-agar during 10 or 14 days. After that we can see colony R or S form colony, and for identification we used biochemical test for ability of microorganism Candida albicans and another species Candida tropicalis and another species for enzymes activity, ability to ferment of carbohydrate. Serological technique, reaction agglutination, reaction complement fixation reaction immunofluorescence we use for detection of antigens Candida and antibody in bloodstream of your patients against antigens Candida. We can use intradermal allergic test with candidin and a molecular genetic technique for detection genetic material of Candida. We use PCR technique. For treatment, all Candida strains are sensitive to nystatin, but acid the pupil adsorbed from the jet. It is not useful in systemic disease. Amphotericin B, 5-fluorocytosine, and chloramazole may be used for disseminated candidosis. Now our lecture is finished. Today you can be free, and don't forget take the computer test in your personal account. Thank you for your attention.